Thank you, Dr. Rourke. We're just going to take a minute or two to get the next talk set up. Okay, our next speaker is Dr. Kelly Zamudio. Dr. Kelly Zamudio is a professor in the Department of Integrative Biology at the University of Texas at Austin and an endowed fellow of the Doherty Regents Chair in Molecular Biology. She received her bachelor's from UC Berkeley in zoology in 1991 and her PhD from the University of Washington, Seattle in 1996. She worked at Cornell from 1999 to 2021, where she became a distinguished professor in the College of Arts and Sciences. Her research focuses on the origin and maintenance of vertebrate biodiversity, evolutionary diversification, speciation, and conservation genetics of primarily New World reptiles and amphibians. Her work has been at the forefront of understanding how chytrid fungus has affected frog populations, both now and in the past. In addition, her integrative studies of amphibian diversification, incorporating phylogeographic, genomic, bioinformatic, and ecological data have become a model of how evolutionary diversification is studied on landscape scales. She integrates field research in population biology, demography and landscape and habitat change with laboratory research on the genomic underpinnings of population diversification, speciation, and conservation genetics. Everyone, please welcome Dr. Kelly Zamudio.
Okay, good morning, everyone. It's great to see you here. And I want to especially thank the students and the organizers for having me here. I am really, really flattered to be a speaker at this EIS event. And it's clear already, just in the time we've been here as speakers, that this is all about you guys, the students, and that is just outstanding. So thank you so much for inviting me. Okay, what I decided to do today is to give you sort of a broader view of the kinds of things that we're interested in. We do quite a lot of different things in the lab. And so I'm going to focus on three different types of projects. First, I'll tell you a little bit about some work we've been doing on sexual selection and reproductive modes and mating systems in frogs. Then I'll pivot to tell you a little bit about our disease ecology work on frogs, studying the chytrid fungus, the amphibian killing pathogen of frogs. And then at the very end, very short little segment, if we have time, I'll tell you about using eDNA and some of the habitats where those frogs have gone missing to see if we can rediscover any of them. So let's start with this. This is the segment on mating systems and reproductive modes in frogs. So let me give you a little history. So this all started for me about 10 years ago when this paper was published. Gomez Mestre et al. In 2013 published this paper in Evolution. And I immediately was attracted to it because it had to do about reproduction in frogs. And I am fascinated with frog reproduction. And it turns out that what they did was take the entire phylogeny of all frogs of the world. So it was a really wonderful data set. And they categorized every species according to these traits they were interested in. Then they put out there as their overall goal that they were going to look across the phylogeny of frogs and ask questions about what is it that's driving diversification of reproductive modes. Interesting, I read this paper, it was great. We read it in lab group. I thought, wow, this is awesome, what a database. And then I sat on it and something started bugging me, and then I was just like, what is bugging me about this? So let me just tell you what they found, and then I'll come back to what was bugging me. They, in their discussion, talk about the fact that you see more reproductive diversity in the tropics. Well, we already knew that, but they showed that on the phylogeny, so that was cool. They posit that humidity and temperature limit the evolution of more terrestrial modes of reproduction in frogs in the temperate zone. Okay. And then their big outcome was that over and over in the phylogeny of frogs, the evolution of more terrestrial modes of reproduction, taking your eggs and your tadpoles onto the land and out of the water, was driven by this need to escape predation in aquatic habitats. And that is what bugged me, because if you look at the way that they categorize their traits, they already a priori linked two life stages in such a way that they were already biasing their inferences towards avoidance of predation in aquatic habitats. And what I mean by that is that they categorized frogs as either having both eggs and larvae unprotected, protected eggs, but not protected larvae, protected eggs and larvae. So you see the point, like everything was already set up in terms of the idea that all of this diversity had to do with protection from predation. So those of us who study sexual selection, me and my collaborators, a couple of us, got interested in thinking more about this. And we decided, why does it all have to be about protection? Why can't there be some impact here about the fact that there's sexual selection, there are males making decisions, there are males competing, there are females making decisions, right? And the reason we thought about this is because if you unpair the egg stage from the larval stage in a tadpole, the egg stage, the point at which eggs get fertilized during amplexus in frogs, is actually the critical stage in terms of this selective event, right? Sexual selection. And we know from just looking at frog mating systems, there are a huge number of systems where the dynamics of what goes on right during that critical time, the time of fertilization, the time of amplexus, could favor a lot of competition between males who are competing to fertilize the eggs of the same female, competition against females, competition between females for selection of their mates, and so forth. So here's some examples of those complex systems. We have satellites or sneaker males. This right here is the Great Plains toad, and you can see there's a calling male right there, and then there's a little silent satellite male just waiting for a female to come in. We also have some more extreme examples. This is a leptodactylid from Mato Grosso in Brazil. This is a terrestrial nest that one male has built. Here he is amplecting the female, and all these little males around in a circle are kicking up their sperm into the same basin. A lot of competition here for sperm, for fertilization of eggs. And then you have a number of systems like this one and this hylid, where you have these mating balls, and somewhere in the middle there is a female ovipositing. So all of this led us to believe, well, okay, there's got to be competition here for something. Maybe polyandry, the possibility of actually losing fertilizations to other males, might be contributing to some of this diversity. So to actually test this, we have to do a couple things. First, we need to uncouple the point of amplexus from the development of the larvae. And we need to be able to show that there's a correlation between going terrestrial as a frog, making a choice of where to reproduce, going terrestrial and actually protecting yourself from this exposure to other males, to polyandry. So what I mean by that, just to be clear, is that for example, both of these species here are terrestrial breeders. They're considered terrestrial breeders. This hylid right here has dug a little basin at the end of a pond and is protecting the eggs. So it's a terrestrial situation. It's outside of the main water body. However, this is what we call an exposed amplexus. This male, when a female comes, is still exposed to other males. Alternatively, this Leptodactylus here builds this volcano nest, calls from within it, attracts the female in there, and has exclusive access to the female at the time of amplexus. Okay, so this is what we're calling hidden or concealed amplexus. Okay, so both terrestrial, but different degrees of concealment. So to test this hypothesis that sexual selection has anything to do with reproductive modes, we would have to show that correlation between terrestrial reproduction and hidden amplexus is actually there, that it's a thing. And ideally, we would be able to show that going to a concealed amplexus actually leads directly to a reduction in the competition for fertilization, a reduction in the rate of polyandry. Okay, so we tried to do that. We did this in a couple families, the hylids and the leptodactylids. I'm just going to tell you about the leptodactylid case now. But basically, we go about this like this: reconstruct the phylogeny for the group we're interested in. use the traits that we're interested in, terrestrial or non-terrestrial reproduction, hidden or non-hidden amplexus, and reconstruct the ancestral states, and then look across the phylogeny for correlation between character states for those different traits of interest. And you can do that for all the traits, right? You can do it for tadpole development and egg placement. You can do it for egg placement and whether there's parental care or not, and so forth. So we did that. And sure enough, the only, at least for leptodactylids, the only case of correlated character evolution that we found was between where the eggs were placed, the site for amplexus, and whether the amplexus was exposed or hidden, okay? And in fact, those two traits evolve dependently on the phylogeny of both hylids and leptodactylids. And you can reconstruct the correlation of those traits in this particular analysis here, which I'll walk you through, okay? So this here shows every single possible combination of the two traits that are evolving dependently: aquatic egg, terrestrial egg, exposed and hidden, and they're in every possible combination, okay? The arrows here that are solid are significant. That means that there is a correlated evolution or a correlated change across the phylogeny in those two traits. And then the arrows that are dashed are not significant, so you can ignore them, okay? The thickness of the arrow actually shows you the effect size, like how correlated transitions across the phylogeny in those two traits are. So the way to look at this is to look at these by pair. So let's see up here. Here we have cases of exposed amplexus and then transitions from terrestrial to aquatic egg. And there is an asymmetry. There's only significance in one direction, okay? So what this is telling you is that across the phylogeny as a whole, when we have exposed amplexus situations, nodes that involve exposed amplexus, the tendency is for there to be a transition from a terrestrial egg to an aquatic one, okay? The opposite is true here. If we look at hidden amplexus, in this case, across the phylogeny, across nodes that involve species with hidden amplexus, we have an asymmetry in the correlated trait, which is that aquatic eggs tend to become terrestrial. And then here we have across terrestrial eggs, two significant arrows, and basically we have similar rates of evolution in terms of exposed amplexus and hidden in terrestrial eggs, okay? So what this is showing overall is that hidden amplexus is strongly correlated with terrestrial eggs, at least in leptodactylids, and that exposed amplexus is correlated with aquatic eggs. What we predicted, right? You go terrestrial to be hidden. By separating, I think what happened here is by separating the amplexus point from, you can notice that I've said nothing here about where the tadpoles are. We were able to find this pattern of this association that was hidden before by pairing them together. Okay, so phylogenetically, at least it looks like that supports our prediction that sexual selection has something to do with this. Perhaps escape from polyandry might be involved. What about actually testing this directly with polyandry? So polyandry, as you might imagine, those of you who work especially on organisms that lay a lot of eggs, we all know that it's really, really difficult to measure. It's underreported. Actually doing genetic paternity analyses on some of these organisms is quite difficult, but luckily we have a proxy. And that proxy is the relative size of the testis relative to the body weight of the male. And the reason that that's a proxy for organism is because organisms challenged with, for example, larger sperm competition are in theory supposed to have larger testis size for maximizing production of sperm, right? So we decided to see whether we could find any pattern based on testis size in leptodactylis that would support our hypothesis that polyandry actually occurs more in exposed amplexus and species with exposed amplexus. Thus far, we've managed to get 94 different testes measurements for leptodactylids. There are about 200 leptodactylids, so we're not even halfway there, but we're working on it. This isn't enough to put this on the tree of leptodactylids, so I'm just going to be reporting some raw data. We're working on getting this tested phylogenetically, and we use percent of male weight as the standardized score. So here's a museum specimen from which we've excised the two testes, and you can actually see the testis. It's just a simple measure of the testis weight relative to the weight of the adult. This, in fact, is that species that I showed you, and I'll show you in a bit. This is actually a relatively large testis size for that size frog. Okay, so here's what we found. Frogs with hidden amplexus on this side right here, there are about 30 points right here, all on top of each other, have consistently smaller testis size than frogs with exposed amplexus. However, you will notice that frogs with exposed amplexus have a huge variance. There's actually some species down here with relatively small testes size, but there's also species with very, very large testis size. So this is a 4.5% weight of the adult. That's a huge investment in testicular tissue. A lot of variation here. So they're different overall in terms of means, but some of the more interesting cases, I think, are actually going to be right here, right? Because these are species that have exposed amplexus and yet have relatively small testes. And what that means is that there must be something else going on there. And if we look at these species, what's going on is that these are the species that are highly, highly territorial. So they're highly aggressive. They protect females from invasions by other males and so on. So instead of investing in testes, they're actually investing in other aspects, armament or, fighting power, body size, etc. Okay, so all of this to say that it might not all be about escaping predation. It's not all about getting your eggs and your tadpoles out of water. And it probably isn't. That makes sense to me. To test this, we would need a lot of species-specific observations about what exactly is happening at the level of selection on males and females during these critical periods. And this threw me into this phase of my career where I basically decided I was going to try to figure out every weird mating system in frogs that I could. And I did do quite a lot of that. We looked at species that have very restricted breeding habitats. So in this case, I was trying to find species where the resource was so rare that we would have higher selection. And in fact, we found some interesting patterns there. I looked at species with really, really high male parental care and its association with parentage there. And we even looked at species where the mating system leads species boundaries to break down. So species where they have really long and complex courtships and are still hybridizing across species, even with very different calls. Most of these were based on paternity analyses using SNPs or microsatellites and kinship analyses of different groups in mating aggregations. But the one I want to tell you about is this one, because I think this one actually got us the closest to understanding how selection might be acting differentially on males and females in a breeding situation. So this is the genus Cycloramphus. It is my favorite genus of Brazilian frogs. Cycloramphus comes in two different flavors. There are some species that are saxicolous, like this one over here. They live right at the edge of these high gradient streams and montane regions, and they tend to breed near waterfalls where the spray zone of the waterfalls wets the rocks. And the tadpoles are semi-terrestrial. Once they hatch out of their eggs, the tadpoles crawl up on these waterfall wet rocks and develop by eating algae there. The other flavor of Cycloramphus are the highly terrestrial ones. They also visit streams, but they don't breed there at all. They actually leave the streams, create chambers in the forest floor, and breed hidden. Okay, so in the same genus, you have the two hidden and exposed amplexus forms that I was talking about. You can sort of see the difference in morphology overall. You can sort of see that this is a little bit more fossorial, right? More streamlined, smaller eyes, so on. They spend a lot of time in the forest and in the forest floor. Okay, so the first thing we did was build a phylogeny for Cycloramphus and reconstruct the saxicolous and exposed mating and the terrestrial and concealed mating phenotypes we were interested in. And what we found is that there's been at least 3 different evolutions of this terrestrial reproduction. And you can see them here in orange, these 3 different clades. We can then use this phylogeny and these ancestral reconstructions to ask the same questions about where is selection acting, right? In this case, we focused on body size. This is the reconstruction of female body size and the reconstruction of male body size. And the cooler colors here, as you go to cooler colors, mean larger individuals. There's a general correlation between males and females, as you would predict, which is common. But a couple of things to remember here. Cycloramphus females, as in many frogs, as in most frogs, are larger than the males always. And that's true for all Cycloramphus. So, and we calculated for each species a sexual dimorphism index, meaning the relative size of males to females. High sexual size dimorphism index means females and males are very distinct from each other. Low sexual size dimorphism index means females and males are closer in body size to each other. Our question was, as sexual size dimorphism changes, can we tell whether it's the male that is getting smaller or larger, or is it the female that's getting smaller and larger relative to the other sex? And what does that tell us about the degree of selection and the kind of selection that might be acting on both of the sexes? So there's some predictions that are out there. The female fecundity hypothesis posits that females should always be as large as they can be, right? Females should always be as large as they can be because their body size is directly correlated to fecundity, to the number of eggs. We also predicted that perhaps, given that we had these multiple evolutions of territorial breeding, that perhaps hiding amplexus would have some sort of release in terms of selection on large male size, because the guys who are in the chambers on the forest floor breeding in hidden situations are no longer defending territories at the edge of the stream and at the waterfall. So that's what we were looking for as our main prediction. Okay, so what do we find? Same exact analysis as last time, except this time, if the arrows were not significant, I left them off the graph. And we're looking at the correlation here between terrestrial, which means hidden, or saxicolous, which means exposed reproduction, and the overall sexual size dimorphism index. This is the considered ancestral state from our reconstruction, and that's saxicolous with lower sexual dimorphism index. So you can see here that the male and the female are closer in body size. We have some changes back and forth in sexual dimorphism that are correlated, but not very different from each other when frogs are saxicolous. But the important transition is this one here. There's a correlation between where you live, whether you are saxicolous or terrestrial, and higher sexual size dimorphism index. And that's unidirectional. So basically what this is telling you is when you have high sexual size dimorphism index, meaning the sexes are further apart in size, that those nodes or that part, those transitions in the tree tend to be correlated with a transition from saxicolous to terrestrial. Okay. So we can actually split this apart and see who is doing that change. Here's what we find for males. Now we're looking just at the males, and males that are saxicolous here in blue move back and forth across the tree between being relatively large and relatively small. But the other side is asymmetric, and males that are terrestrial, there's a dependent evolution here between being terrestrial and having transitions that primarily favor smaller males. Okay, so what this is telling us is that terrestriality does seem to be associated, it fits a model of dependent evolution with smaller overall cycloramphus males. If we look at females, there's almost no significant differences. The females tend to be larger, as I said before. There are differences, there are changes that are correlated and significant here in saxicolous species, but they tend to be equal between relatively smaller or relatively larger. So what this means, what we can infer from this, is that these changes in sexual size dimorphism actually come from differential selection, we infer, on males and females. And males, as was predicted by the fecundity hypothesis, tend to stay large, don't do a lot of changing, and it's the females don't do a lot of changing, and it's the males that shrink when you move onto land and hide your amplexus. Presumably because of a release from selection on the need to be large for territorial defense. Cool, huh? It's like exactly what we wanted to see. It was fun. Okay, I'm going to switch now and talk a little bit about some work that we've been doing with the amphibian chytrid fungi. For those of you who don't know, there are two chytrid fungi that are obligate parasites of vertebrates, and specifically of amphibians. One is called Batrachochytrium dendrobatidis. We call it BD for short. The other one is called Batrachochytrium salamandriveran, and we call that BSAL for short. Both of them are really terrible pathogens if you're an amphibian because they really like aquatic and moist habitats. They love keratin. Amphibian skin is full of keratin, and they actually move through aquatic habitats with zoospores that have a little flagella. So they're motile and they can infect from frog to frog or salamander to salamander that way. BD has been known and studied for quite a long time, a couple decades at this point. And BSAL was just described in 2014. It is actually not known to be in the wild in North America yet. And of course, we are an important center of diversity globally for salamander diversity. So there's a lot of eyes on what's going to happen with BSAL. So every time I give a talk about BD, these are the two questions I get. What's the impact on amphibian biodiversity? Like, how bad is this really, right? And then the second question that I get is, where did it come from? Like, what's going on? So I, along with a global network of people, I cannot take credit for this work in any way by myself. Every one of these projects was done with like 100, 200 people involved to actually get the global reach that we were interested in. Actually tried to answer this question, how bad is it really? And so we surveyed every single species in all the countries that we represented and tried to come up with some at least rough estimate of the degree of endangerment of species caused by Batrachochytrium. And it's fairly rough because there are not a lot of good data out there about the impact of this disease specifically, or at least there are not a lot of very precise data. And so what we did was categorize a species as having no effect at all that we could tell, being affected a little bit, maybe 20% reductions, being affected somewhere in the middle between 20 and 90, being affected heavily, meaning 90% reductions in population size, or being presumed extinct. Okay. So here's a global map with all our different endangerment categories. As you go to warmer colors, that means you go from less than 20% impact to presumed extinct and extinct. And what we found is that there are about 500 species for which we have some evidence, either through retrospective museum specimens, reports, gray literatures, specialists, monitoring in the field, et cetera, that there's been an impact on that species to some extent. Okay, so 501 species out of the about 8,000 species of frogs had been impacted by this pathogen in some way, to some extent. So just to give you a context, if we were talking about mammals here and 6.5% of mammals were impacted to some degree by a single pathogen, that would mean all primates would be having some level of decline. Okay, so it is a significant impact on frog biodiversity. We also found some patterns that were not unexpected, but show sort of what happens in terms of emergence of infectious diseases like this. Most of the extinctions and severe declines that we categorized actually happened before BD was even described as a species. This arrow right here shows BD. The 1999, the year BD was discovered. And you can see that there's a lot of red up here before then. If you look at these concentric circles and look at the distribution of colors towards the center, this is range size getting smaller as you go towards the center. And if you squint at it, you can see that most of the warm colors, the highly endangered species, are actually towards the center of the circle. And so species with smaller ranges are more impacted, not surprising. And then also, if you look at elevational range, the range of elevations at which a species occurs along a mountainside, for example, what you can see is that species with a higher decline color are actually narrower in elevational range. So all this says is that if you have small ranges and if you're a habitat specialist, for example, you're going to be more impacted than if you're a generalist and have a large range. And that's what we found. The second question, where did it come from? We basically created a phylogeny for a panel of 234 BD isolates and tried to reconstruct the genetic relationships between them. I'm going to show you the tree here, which is a little complicated, but I'll walk you through this. This is the phylogenetic tree for BD for all these isolates. This is the root of the tree right here. And I just want you to notice two things. The first is that there are these deeply divergent lineages at the base of the tree that have different colors. These are what we call the endemic lineages. They're in particular geographic places, and they tend to be restricted to those particular geographic regions. So in pink here, for example, we have BD Brazil, which is one of the lineages I've been working on in South America. In blue here, we have BD Asia. This is BD Africa. And then this green, very shallowly diverged lineage is what we call BD GPL, the global panzootic lineage. So that's the one that has gone all over the world. And that's the one that actually is hypervirulent compared to the others and has been associated with most of the amphibian crashes that we hear about, like in Central America and in Mexico and in Australia and in California. Okay, so this is the dangerous clade. Think of it that way. Oh, where did it come from? Using this phylogeny and using the distribution of genetic variability across these different clades and across the globe, what we find is that most of the genetic diversity is actually in Asia, and that all the genetic lineages that are found in other continents are actually a subset of the genetic diversity in Asia. So we think that, well, the global panzootic lineage is everywhere now. You can see the green bars. But we think that this points to Asia as a center of origin for the emergence of BD and the emergence of BD GPL, which then secondarily spread globally. Okay, my lab has been focusing on understanding the reaction of hosts to BD. And we do this mostly through differential gene expression studies. And I'm going to tell you about two of them right now. This one right here has to do with organisms that are infected by both of the Batrachochytrium, the frog one and the salamander one. And I'm going to tell you about a study that looks across these different lineages, the BD Brazil lineage and this hypervirulent lineage. Okay, so these are gene expression studies, and I'll try to keep it relatively simple. I'm just going to show you the highlights for each one. But in this study, we actually co-infected salamanders, red spotted newts here, with either one of the pathogens, the other, or both. And the reason we did this is because there are a couple different predictions of what could happen. If these pathogens compete with each other in the skin of the salamander, it's possible that you could have an antagonistic response, and that might even decrease disease severity. However, if these two pathogens interact in a synergistic way, then the salamander really has an uphill battle, right? Then we could actually see an increased disease severity. So we did infection trials with 4 different treatments, BD only, B-Sal only, controls, which were sham infected, and then both pathogens at the same time. We found a number of genes that were differentially expressed. In these volcano plots, every single little dot is a gene that's expressed. If the dot is colored in red, then that gene is overexpressed relative to the control. If it's in blue, then it's down-regulated relative to the control. And our first signal that something was up is that, look at the volcano plot here for the co-infected organisms. There's a lot going on. This is salamanders infected just with BD. This is salamanders infected just with B-Sal, and this is salamanders that were co-infected. Now, just to make clear, our salamanders walk around out there in nature right now, often infected with BD, and they tend to not have a huge problem with it. They're relatively tolerant to BD. So if B-Sal were to arrive here, we would automatically be in a situation where many of our salamanders are going to be co-infected, right? Because they already have BD. They already live with BD. Okay, if we look at the genes that actually are expressed, it's crazy. You see genes that you would expect to be expressed if something is challenged by a pathogen. You see genes having to do with innate immunity, adaptive immunity, everything. On the other side, oh, I just forgot to mention, these right here are the controls in yellow, and these right here are the co-infected individuals. And you can see it's like opposite patterns of gene expression. Okay, so everything that's underexpressed here is overexpressed here. Everything that's overexpressed here is underexpressed here. 3 of the genes that we found that were most differentially expressed have to do with this complement system, which is an important part of the innate immune system. Okay, so what's happening here is that we have a bunch of things that should be upregulated, getting upregulated, but then in infected individuals, they actually happen to be downregulated. Okay, and including things like the adaptive response, which is the immune response that eventually leads to resistance through the development of antibodies. Okay, I looked at some of these genes, or we looked at some of these genes specifically. These are 3 genes just as examples. And here's the control. This is infected with BD only, B cell only, and co-infected. And in every case, it's like this, right? Control has basically normal expression levels, and then you get an increase with BD, an even larger increase with B cell, and in the co-infected, you get a sharp drop in terms of the expression of that particular gene. What this leads us to believe is that there's some sort of downregulation of critical immune genes that has to do with some form of immunosuppression. It could be that this is host-directed immunosuppression, like the host just can't deal with co-infection. And it could be that the pathogens are actually mediating immunosuppression. That is known actually for some other fungal pathogens. So immunosuppression, and then sure enough, these are work by a previous graduate student of mine who's now a professor at University of Florida, and she looked at survival curves for these different treatments. And what she found is that BD alone and the control are up here at relatively high survival, like above 90%. B-Sal alone results in some mortality, but the probability of survival is somewhere on the order of 50%. And if you have BD and B-Sal infecting salamanders, you get tremendous mortality at the level, I mean, basically only a survival of less than 20%. So this does not bode well for salamanders, especially here if we have an invasion of B-Sal. And the mechanism we think has to do with this immunosuppressive effect. Okay, the second question had to do with what happens when hosts are infected by these endemic lineages, these like geographically restricted lineages compared to this hypervirulent thing. What's the mechanism that's causing this lineage to be hypervirulent? So here are predictions. We predict that GPL, the hypervirulent lineage, will result in higher disease severity, and that maybe a species that has co-evolved with a locally restricted lineage like BD Brazil would have less disease severity. So we chose this host. It's called the pumpkin toadlet. It's a species endemic to Brazil. And our question was basically, can we differentiate host responses to this enzootic BD Brazil lineage compared to the hypervirulent one? Okay, here are the survival curves. You can see that BD Brazil here in yellow, everybody kind of survives pretty well. This is the probability. I can't read if it's survival or survival probability. And you can see that everybody's surviving fairly well. And then BD Brazil has a drop here at the end. And actually, if you look at the degree of infection of the pumpkin toadlets, everybody, independent of lineage, in yellow BD Brazil and in purple, in yellow BD GPL and in purple BD Brazil, you could see that everybody gets infected, everybody develops a high load on their skin, but the BD GPL maintain a higher load throughout the infection. Okay, so BD GPL infects and is virulent. BD Brazil also infects, but seems to be dampened in some way, not only in terms of mortality, but also in terms of the total amount of zoospores in the skin. What's going on here? So in this case, we had the opportunity to actually sample at 3 different points in time because we wanted to see the progression of this immune compromise, right? So we sampled early in the infection, mid-infection, and then late in the infection. And what we find is like completely opposite responses to the two different lineages. There are many more genes that are expressed in response to BDGPL that are differentially expressed, and they tend to be differentially expressed really late during the immune response. Okay, so here this just summarizes this. This is the result for the skin. Early on, there's some expression to both. In mid, BD Brazil response kicks in, and BD GPL response only kicks in relatively late. If we look very specifically at what's going on, here's early, mid, and late. The top panel is the response to BD Brazil, and the bottom panel is the response to BD GPL. And so you can see that for pumpkin toadlets in response to BD-GPL definitely are at the end. Oh, I'm sorry, I forgot. These are all different categories of genes that are associated with immune response. We just selected immune genes in this case. You can see that pumpkin toadlets in response to the hypervirulent lineage are actually very early on trying to do some sort of response. That's these light green bars. And then BD Brazil infected frogs actually don't do anything early on. They ramp up their immune system, as you would predict, 6 to 7 days in. And so they have high overexpression of immune genes at the mid stage. And by the time they get to the late stage, they've pretty much resolved their infection. And then what happens is that BD-GPL comes in late. Frogs infected with BD-GPL respond late. So what we think is going on here is that BD Brazil is successfully responding in terms of their immune system. They have epithelial turnover, and they basically have achieved some form of tolerance. Whereas frogs infected with BD-GPL have this delayed immune response. They try early, nothing happens. Then they keep trying mid, and then they keep trying late. And by the time they get to late, the whole thing is magnified, right? So by the time we get to late expression, I think what we're seeing here is just the host throwing everything it can at the fact that it's not able to control this particular infection. So again, this is a signature, a classic signature of immunosuppression. Again, we don't know the mechanism, but likely motivated by the pathogen itself, or potentially motivated by the pathogen itself. All right. So that's BD. I don't want to leave you with this terrible immunosuppression story that is depressing. So I'm going to tell you a little bit about some of our efforts in the field to try to rediscover some of the species that have gone missing, some of them potentially in response to BD. So we did this work in Brazil, mostly in the Atlantic coastal forest. It's a biodiversity hotspot for frogs. Many of the frogs in Brazil live in the Atlantic coastal forest. And you can see that the Atlantic coastal forest is vastly shrunk in size. Approximately only 16% of its original cover remain. However, there's still a lot of frogs there. And the interesting thing is that early on, back in the 1970s, people started calling alarms about the fact that a number of frog species were going missing. This was before we knew about BD. And I'm not saying that all these frogs have disappeared only because of BD. In fact, that's not the case at all. I'm just pointing out that there are multiple stresses on frogs in this area. But this is what the literature said. Like, we look over and over, drastic population declines, general decline of frog populations, stories of, these yellow jewels used to carpet the floor of the forest, and now we go and we find 1 or 2 and so on. So 5 species have become extinct. It's all over the literature in the 1970s. So what we decided to do was to glean the literature for all the species that had gone missing. And we found 30 species that seemed like good targets. And they were spread over 6 different localities. And we categorized on purpose those 30 species into 3 different endangerment categories. So for some species, we had reported declines, but they were still present, even though at low density. For other species, they were completely locally extinct. They were locally extirpated, but existed, we knew, in other parts of the range. And then the third category, which was the most endangered, was the one where it's disappeared and it's disappeared throughout its range. Like we can't find it anymore, okay? So those 30 species are roughly categorized into those 3 endangerment categories. We sampled water from each of these localities. I say this is like the most beautiful field work I've done, but also the most boring because it is in these beautiful places and we're sampling water at the edge of these beautiful clear streams and there's not a single frog to be seen. But we filtered liters and liters of water at each locality, about 60 liters per cartridge. You capture the DNA on the cartridge, do a whole DNA extraction, and then we basically meta-barcode that extract to look for signatures of the different species that we were looking for. Now, we were looking for 30 different species, but we took a slightly different approach than most eDNA studies in that instead of targeting specific species with a marker or a barcode that we knew matched only that species, we actually tried to identify every frog species that was in our cartridge. And we did that by creating a sequence reference database that was very extensive. The reason we did this is because a number of the species in our list of 30 were actually presumed extinct, and we don't know what their sequences look like, right? And so we were trying to basically, using detective work, get closer and closer and closer to a species identification without having the sequence for that particular species. So in this reference database are all the species we were looking for. If the species sequence did not exist, we put every congener, every species in that same genus into the reference database. And then, of course, we categorized those to take into account geographic variation. We actually put in, when we could, sequences from the type locality and sequences from other localities throughout the range, okay? All right, so let me show you what we did. We did this at all different localities. I'm just going to show you a couple. So this was Estação Biológica Boracéia. We had 12 eDNA samples from this locality. We found 40 OTUs, molecular OTUs, meaning variants that we could assign to frog. Of those, we could identify 21 different taxa, at least to genus, and none of them matched the species we were looking for at Boracéia. Here at Itatiaia, a national park in Rio de Janeiro, we had 16 environmental DNA samples, 20 OTUs. We could identify 13 frog taxa, at least to genus, and 2 of those were the ones we were looking for, 2 species of Hylodes. And then here at Serra da Bocaina, this was very exciting. We had 5 DNA samples from a single waterfall, which was the type locality for the species we were looking for. We had 19 OTUs that were identified as frog. We had 19 OTUs, 15 that we could identify down to genus, and 1 of them we could take all the way down to the genus Megalosia. Megalosia bocainensis was presumed extinct from this locality in Bocaina, and it wasn't just 1 strand of DNA. We had multiple capsules and multiple samples that identified Megalosia. Now, we don't have a sequence of Megalosia bocainensis because it's presumed extinct, but the closest match to Megalosia bocainensis was a different species of Megalosia from a nearby range. Okay, it's of a different species. So we're inferring that this could be Megalosia bocainensis unless some other new Megalosia has moved into this range. Okay, so this is what we found. Of the 30 species, we found 7, which are pictured here. And the important column to look at here is the eDNA. And the ones in green are the ones we found. And as you can see, it sort of matches what you would predict. 4 of the 7 we found were in the declining category, meaning they were there, but in potentially very low density. 2 of them were in the locally disappeared category, meaning they were thought to be locally extirpated, but apparently are still there at densities that are not identifiable. And then we have 1, the one I just told you about, that is Megalosia bocainensis that was in the endangered, in the extirpated category, in the completely disappeared category. Well, is this a success? That is for you guys to help us decide. Of course, finding strands of DNA in beautiful streams is not telling us anything about the health of a population or the potential for recovery. But in my opinion, it actually does give us something to look towards, right? If not at those particular localities, which were the type localities, then maybe at localities in the same drainages or streams in the vicinity that might still be refuges for populations that were thought to be disappeared. All right, I'm going to end here. I just want to finish off by telling you guys that I hope I've convinced you that looking at traits, environments, and function independent of the context can actually have offer important inferences or important information about mechanisms of diversification and also the conservation of organisms. And I want to thank all my collaborators in 3 different areas and my funding sources, and I'm happy to take any questions.